Hello, everyone. Thank you for joining us today. My name is Devin Simmons, the STEM Lyceums Program Manager, and I am thrilled to present to you our Science and Music webinar sponsored by the Claude Moore Charitable Foundation. The Center for Excellence in Education was founded in 1983 by Joanne DeGennaro and the late Admiral H.G. Rickover, who is the father of the nuclear navy and civilian uses of nuclear power. CEE's mission is to nurture high school and university scholars to careers of excellence and leadership in science, technology, engineering, and mathematics, and encourages collaboration between and among scientific and technological leaders in the global community. You may visit CEE's website to learn more about the center's cost-free programs for students and teachers, such as the Research Science Institute, USA Bio-Olympiad, Teacher Enrichment Program, or TEP, and STEM Lyceums. And for the big part of our show, we are pleased to have an extremely insightful speaker for today's webinar, Dr. Mark Temple. Dr. Mark Temple is a senior lecturer in molecular biology at Western Sydney University, is renowned for his expertise in chromatin structure, anti-cancer drug interactions with DNA, and his investigations into aging using yeast models. His recent pioneering work in applying audio for biological sequence data and analysis has resulted in the development of DNAsonification.org, including the groundbreaking sonification of the coronavirus genome. Temple's innovative fusion of science and art is evident in his successful events such as This Sounds Like Science and Science Meets Art, highlighting the harmonious blend of scientific exploration with artistic creativity and expression. Dr. Mark Temple earned his PhD from the University of New South Wales. And without further ado, Dr. Mark Temple, he's going to take it away and share his groundbreaking knowledge. Thank you, Dr. Temple.
As Devin was saying, my name's Mark Temple. I come from the School of Science at Western Sydney University. And I don't have an Australian accent because I grew up in England. And I came to Australia about when I was about 18. So enough of my life story, but that's part of what's happening now. I was amused when Devin referred to me as the renowned Mark Temple, only because I think on the weekend I was at a science fair, and again I was referred to as the renowned Mark Temple twice. So I've only been referred to as renowned twice in my life, both within this window of about 4 days. So maybe it's just one of these things that happen as you get older. Okay, so I'm going to talk to you a little about, I guess, my journey through science, and a little bit about how I've managed to pull together my interest in the arts together with my interests in science. And it's been, I wouldn't say it's something I planned as a career. I would say it's something that has just naturally emerged from having strong interests in both domains. So if I was to tell you that I planned to do what I'm doing, that would not be correct. It's just, I've just maintained an interest in the things, I've maintained a focus on the things that interest me. Okay, so let's move forward. All right, so at the top of each slide here, I've got a little prompt which comes from Devin's questions to me. So it's a narrative I'm following, which is, I'm trying to follow a certain narrative here. So forgive me sometimes if I'm reading the top of the slide as you are, to say, now what's this all about? So as an introduction to me, a bit of a background on my working with science and music. I think it's not difficult to understand how people have an interest in music. I think most people have an interest in music, and that goes without saying. Having an interest in science is something you either do or you don't. I think the more you want to understand the world around you and the more you want to be able to think clearly about how things work, then science is a great segue into that. So I've got two little slides here. One on the right is my science world. So, obviously I went to uni, I did an MSc, I did a PhD, I did some postdocing, and then I went and become an academic. So I followed that linear path, which is pretty much laid out for you. If you do well at uni, you have the option to stay at uni and not go into the real world like I did and become an academic. All right. So here's an example of one of my publications. And there's no need to read the title or worry about what it means, but the keywords are, I was doing stuff with DNA. I was very much focused on DNA, so at the bench, I was getting cells, pulling DNA out, and I was adding anti-cancer drugs to the DNA and looking at where they bind, what happens, and all that, So I did that for a few years, and I thoroughly enjoyed my PhD. That was one of the really great time in my life to be doing my own science at my own pace with a bunch of really motivated people in the lab and a great mentor. It was absolute great thing to be doing. Before I did the science, because I ended up going into science as a mature age student. And I did that because I had a misspent youth. And that misspent youth was me doing music. So after my undergraduate, I got to the situation where I had university demands saying, do this experiment, do this homework, do this, do that. And that was one voice shouting at me. The other voice shouting at me was, I was playing in a band and we were getting more and more people saying, can you play this show? Can you go to Brisbane? Can you go to Melbourne? Can you do some recording? Can you come and play on the radio? Can you do this? Can you do that? So I had these two voices shouting at me and demanding my time. And even though I kind of loved science, I saw the music as an opportunity that might not be there again. Whereas I saw science as like a linear pathway, which hopefully will be there for me at some stage. So I dropped science and I grabbed this music opportunity. So I think of it like running away to join the circus. I sort of left all my training behind and went and did music. So on the right here, I've got a little snippet of well, it wasn't our first single, but it was our first commercial single. So after leaving science, within about a year, I'd arrived in this place. So I'll just quickly play a bit of this. Just for those of you who can't recognize me now, this dude here on the bottom right with black hair is me. So I haven't got the black hair anymore, and I've got these glasses and everything. Anyway, here we go. I'll just see if this plays. She said she was leaving. I said I don't believe it. She said it, but she I like this bit here where I'm jumping around like an idiot with my friends. That's something that you don't get the opportunity to do that very often unless you're at a dance party. All right. So that's a bit of a, so in my world, I had two competing interests, and I couldn't do science and music simultaneously. And I didn't really think about it at the time. Like, I was happy to be doing either science or music because, that's just what I was doing. After about, maybe 6 years, I mean, we were a professional music band. We had top hit singles in Australia. We toured, we did a festival tour of Europe with, The Cure and all these other bands. We played lots of, played in front of 350,000 people in, Austria, and we played Glastonbury in England, and we did all that kind of rock and roll stuff. Then we went to America, and we recorded our second album in North Carolina, and we did a East Coast, West Coast American tour. We did all that sort of stuff. But at some point, I was scratching my head thinking, in 20 years' time, I can't see myself playing drums in a rock and roll band. I need to think about how I'm going to get buy a house or, have a relationship, So I didn't see bouncing around, in a rock and roll band as a sustainable way of life. Now, that was probably good or bad thinking on my behalf, I don't know. But I made the decision to leave the band and go back to science after about 5 years. So to jump ahead now, maybe 25 years, what we've got on the slide is where I've ended up. And I'm really kind of happy with what I've produced within the science domain here, because I've now reached a point where I can do science and music simultaneously. I've kind of harmonized these bits of my brain that I wanted to rush off in different directions into something that's more focused. So let me just play you a little of this, and then once I've given you this context of what I've created, I can then talk about the hows and the whys and the science behind it. But just to try and get you interested in this, I'll just play a bit of it and then explain it to you. Okay, so I'll play a bit more of that later, but what you can see here is something that almost sounds musical. For those of you who remember Game Boys, then you might recognize that do kind of sound. So it's just really low bit rate audio. So what I'm doing here is using audio for DNA sequence analysis. All right, I'll say that again. I'm using audio for DNA sequence analysis. And this is something that's called sonification, which is not a really pleasant word. So I sonify things. So there's lots of examples where people use audio to tell them about data. And I'll give some examples of that later. So as a scientist, I've moved on from doing benchwork experiments, adding anti-cancer drugs to DNA. And I've started to use some sort of coding abilities to do DNA sequence analysis. And I'll explain to you the process in a short while, but here's two examples of some papers that I put out sort of semi-recently. So the first one is an auditory display for DNA sequence analysis. So when you think about an auditory display, it's different from a visual display. So it's really straightforward. You can use your eyes to perceive data, or you can use your ears to perceive data. And what I'm arguing in this paper here is a proof of principle that audio is useful for DNA sequence analysis. Okay, and then jump ahead a couple of years, the coronavirus lockdown. I was at home for a few months, and what I did during that period, my COVID project wasn't sort of growing vegetables in my back garden, or that kind of, or joining a book club. It was developing a visual display of the coronavirus genome, which is what I was playing in that previous slide. Was the audio display. It's like an audio and visual display of the coronavirus genome. So I'll talk about the science behind what's going on to generate the music, and then we can have a think about whether that music is actually audio or is it music. Is it scientifically useful, or is it just Game Boy music? And those kinds of questions. So if you take this process of being a scientist, using audio for science analysis, because I was also, probably still am, a musician, there was a joke going around the music community is, what do you call people who hang around musicians? And the answer would be drummers. But I kind of think that drummers can be musicians as well. So I'm gonna put one on the board here for a drummer being a musician. So the logical consequence of using audio for data analysis is to think about the audio as music. And then if it is music, then can you play with other musicians to the audio? And I think the answer, at least in my world, is yes. So here's a little performance here done during National Science Week. So in Australia, in August, we have a week, or 2 weekends and a week, so 9 days, of popular science activities all around Australia, in the main cities and out in the country. And in this one here, I did, I played music to the coronavirus genome. Or actually, this one is to the myrtle rust genome. So I'll just play you a snippet here. There's the sound of the DNA sequence pulsing away. Okay, so in this context, what we're doing, we're just playing the sequence in the background. And, don't forget the sequence, it isn't music in the sense that it doesn't have an introduction, doesn't have a middle eight, it doesn't have an ending, it doesn't have a crescendo. It's just the DNA sequence pulsing away, telling you about what's happening on the DNA level. But we then just jam and add a little bit of, jazz style here, indie rock style here, and just general music over the DNA. And these are really great opportunities to reach out to the public and say to the public, here's some information about how cells work. Here's DNA, here's polymerases, here's gene expression. And we talk about a little bit of basic science, but then we also talk about issues like the coronavirus and the myrtle rust, which I won't go into today. And then we do a little bit of music performance based on science data. And it's a really great way to engage people. It's a really great way to open up conversations. And yeah, so that's what I've been doing. So, all right, so getting back to the narrative here, so I don't go too much off topic. A little bit of background information about how the science influences the music and the music influences the science. So I think I've kind of talked about that, about how the two are complementary in what I'm doing. So like I said, I'm using audio for data analysis. And then why and how useful is it? Well, I'm trying to show in a proof of principle of whether you can detect a gene sequence from a non-gene sequence, or whether you can detect repetitive DNA sequences from complex DNA sequences. And you can, and I can show you some examples of how clearly you can hear those if you like. So now, does being musical help me develop the tools for DNA analysis? And I think the answer is yes. And what I'll do to try and show you how the music has informed my science, I'll give some early examples of sonification and some later examples of sonification. So you've got science sonification without much music theory, and then you've got science sonification with music theory added. And the difference in terms of what you can get from the audio, I think is really interesting. So like I said, the audio is not music, okay? It's not intended to be music, all right? But it can be made into music. So let's not confuse science audio with music. And the sort of journey it's taken me down has been also to start thinking about generative music, but I'll see if I get to that towards the end of the slide project, the end of the talk. So now I'm trying to think about the science and the audio and how it kind of works together. So I'm a molecular biologist, but I also do what's called bioinformatics, which again is this kind of scary word. It's just, like computer biology. Using computers for data analysis is quite straightforward. So I take biological data. Now, I could have taken stock market data or any kind of data, but I'm a biologist, so I take biological data, such as a DNA sequence, GATC, just a long sequence of bases that we need to understand. And I put it into a black box, which is my sonification tool. Or I put it into a black box online, such as a genome browser, and it generates a visual display. Now that's generally how we do data analysis, is through visual displays, and we can perceive these things. My kind of research has added audio displays to the mix here, and I think that's really powerful. But in isolation, I don't think the audio displays are enough. So a combination of a visual display with an audio display, I think, is really good for perceiving data. So we've got our data, we run it through these black boxes. We know that the visual thing is always going to be the same. If I've got one sequence and I visualize it, it's going to look a certain way. And the audio is the same. The audio will always sound the same based on a DNA input. So it's systematic. It's not generative. Generative means the music kind of evolves and creates its own sound that you can't predict. That's not what's happening here. This audio is systematic. The same DNA produces the same sound, the same audio, okay? So it's just as a quick primer on what is DNA, because I spend a lot of my time looking at DNA. I've spent 20 years focusing on DNA. Here's a three-dimensional structure of DNA, and it's kind of a molecule that runs in that direction, and it's two strands running in opposite directions, and they twist around each other. So it's we call it a double helix. Okay, so I hope that didn't shock you, that audio then. So, well, what's that? So that was my initial sonification, the very robotic, very sort of precise bang, bang, bang generation of audio from that DNA sequence. And I just spun it round just to make it look nice. Okay, so what was going on there? What I'm trying to do, again, if you look along the bottom here, this pink straight line is a DNA sequence. Okay, it's the double-stranded DNA. As a biologist, we want to know about the function of DNA, all right? And the function of DNA is that we've got our DNA, proteins bind to it, and then read along the DNA. It's like a train running down a railway track, okay? So you've got these proteins shooting along the DNA. Why are they doing that? As they read through the DNA, they produce copies of the DNA that are then translated into proteins. So this is called gene expression. And I think a lot about that process of how do we express a gene into a protein. And what I've done, I've copied the rules that biology uses to make proteins, and I make audio. So there's a process that reads DNA to make proteins. I copy that process to read DNA, but instead of making lots of amino acids, which are joined together in a protein, I make lots of audio notes, which are joined together to make music or audio. So I've got a really silly animation here. So here's my bit of DNA running across the page. This is a polymerase. It's a big protein. And as it reads along the DNA, it will make what we call a messenger RNA. So I'll just quickly run my animation. So as this protein moved across the DNA, this thing was made. All right, so I'm copying that process with audio. All right, so what we have a movement over time. So when we're doing audio, we need to have music occurring over time. So I'm imitating that process to make music. So at each stage, as we're reading the sequence here, I'm sonifying it to create a musical note, okay, which is making audio for data analysis. So this is the biological process I'm talking about. It's called gene expression. So we convert DNA into proteins, and the processes are called transcription and translation. And that's biology 101. If you haven't studied it at school, if you do science, you'll study it in first year university. It's a pretty well understood process that's been in the literature for 40 years, okay? So what I'm doing on the right here is copying that process. But I'm using algorithms and computers rather than, you know, things in cells, things in our body. And instead of making a run of amino acids, such as methionine, cysteine, serine, valine, you know, cytosine, all of these amino acids, I'm making notes of music, okay? And then when we look at the notes here, it tells us what was in the DNA. In the same way as that if we know a protein sequence, we can work backwards to work out what the DNA sequence was, and also what the function of that DNA, because the function of that DNA must have been to code for a gene. So, you know, so I generate the audio, which tells us about the DNA. So this is the DNA sequence I was showing you earlier, and I played you that audio from the DNA. So what we're looking at here is, and I've taken one of the strands, there are two strands here, and I've just indicated the nucleotide bases that make DNA. So DNA is a long polymer of repeating units, all right? And so for instance, here we've got A, T, G, C, C, A, T. And all of these kind of are the names of the chemicals that we've joined together to make one strand of DNA. So what I do, I take that sequence of DNA and I imagine I'm a protein, like polymerase, kind of reading through the DNA and telling me about the sequence, getting the audio to tell me about it. So what I do, for argument's sake, I take a group of 3 bases here, and that's what happens in cells. We have the genetic code reads 3 bases at a time, and I take those 3 bases and I make an audio note from those 3 bases. I then imagine the protein moving along the DNA to the next note, and then the next one. So as we're reading across the DNA, we're saying, well, what's in the sequence? Tell me using audio. So at its most rudimentary level, that is sonification of DNA. Okay, and I'll explain how I've kind of developed these ideas. Another thing to know about DNA is that we talk about open reading frames, and open reading frames are the same as gene sequences. Okay, but because we look at 3 base pairs at a time, how does the cell know whether, you know, if you've got 3 bases here, should we start reading on the first base? Should we start reading on the second base? Or should we start reading on the 3rd base? Okay, when we get to the 4th base, it's like being on the 1st base, because we've got a sliding window of 3 at a time. So we'd go, you know. So anyway, there were 3 ways to read one DNA sequence. So again, how do we show that in audio? Here's my example here. So there's the first reading frame. It's the second reading frame. It's the 3rd reading frame, and then the 4th reading frame back to the first one. So that sounds like this. So we're starting to layer audio into the audio display here. So adding more data to what we're hearing and starting to think about, well, what's happening at the level of the DNA. So my first sonification project is shown here. So let me just click this link and close the window. So this is my first attempt at sonification of DNA. The interface is not particularly useful, not particularly nice, but anyway, so let's sonify at the most rudimentary level, just those individual bases. And I'll just choose a random sequence, which has just happened here. So I'm sonifying this sequence here. Okay, and it sounds like this. Well, so it's not musical. It's really annoying to listen to, and all it's doing is saying every time there's an A, play this note. Every time there's a T, play this note. And what I did, I came up with 6 algorithms. Each one played individually, and then you could just listen to different aspects. So that was listening to the codons, that are the amino acid residues, or you could listen to, you know, pairs of dinucleotides. You could listen to different aspects of the DNA, and everything was done independently, and you couldn't layer them together. And the reason for that is when I tried to play more than one stream of audio from the data, it just sounded so chaotic that it was unlistenable. So in my science world, I was using audio for DNA sequence analysis, and it was, you could listen to bits of information, but when you tried to add more information into the audio, it was just chaotic. And that's when I had to really think more as a musician to bring musical ideas into my science so that I could layer the audio and still kind of perceive what's happening at the level of the DNA. So as I alluded to earlier, during coronavirus lockdown, I did version 2 of my sonification tools, and I came up with this. So this is another website that has some visuals to help you understand what's happening with the audio. And it has layers of audio. So I'll start playing it, but then what I'll do, this is like a mixing desk here. I'll strip out some of the audio. Well, let's start simple. Let's definitely turn off all of these streams of audio here and just play the single nucleotide. So let's just have a quick listen to that. So what we're listening to here is this. We've got 4 bases, A, T, G, or C, and we're hearing one of 4 notes, okay? Another way of sonifying DNA is to listen to the protein sequence, okay? So every time, so these sort of proteins at the top here is what we're listening to. Another thing we can listen to is what we call the GC content. And it's just a calculation from the DNA. So from the sequence of the DNA now, I've got those multiple layers of audio, which we'd listen to in isolation, but now I can start to layer them because I've thought more about the music and how it, if it was music, how would you sort of construct this into a piece rather than just random bits of DNA or audio. And it starts to sound like this when you layer it together. So you've got, say, every second bass is doing that, but at a quicker rate, you've got this. And then if you look at the GC content, which is something we do in biology, and then if you look at the actual DNA, the protein sequence that we're generating, I'll add that. I won't bother adding the rest of them for now. So by thinking about the process of using audio for DNA analysis, by bringing in my musical sort of brain and starting to think about how do you, let's generate root notes and fifths, and I'll do that in 2 octaves for the pulsating beats that sets up the scale. And then when I've got things that are unusual, I'll use a higher pitch and a more, abrasive instrument to go, oh, what's that? And then when you get to the start of a gene, I'll turn on the audio for that sequence. And when I get to the end of a gene, I'll turn off the audio for that sequence. So it's this great mix of science and audio and music thinking to create something. And that's generated a lot of interest within the science community. So after I published my my paper, within a week, I had Nature phoning me up on the phone saying, tell me about this thing you've just created. And they did, and I got, it's just become crazy since then. So anyway, so I'm using pitch and audio, which is my musical thinking, to make the data analysis more clear. And I'll get to this. I don't know whether I should talk more about the raw science audio or the music. What I'll do, I'll quickly give you one example from science audio, and then I'll give you one example from what I was doing on the weekend, which is this slide here.
And Dr. Temple, you have about 3 more minutes before we go into our question and answer portion.
Okay, I'll skip talking about what I've been. Let me talk about what I was doing on the weekend. Okay, so had a crazy weekend. On Saturday, Sunday, I went to Portland, which is country New South Wales, about 3 hours drive, and I went into this old boiler house, and I set up my equipment in the boiler house, and I ran an installation, and members of the public would come in and talk to me about what's happening. So what we've got here, I've got the DNA audio pulsating in the background, and as a completely other thing that I haven't told you about, I started playing around with generative music, but it's generative music from plants, okay, which is basically detecting voltage differences in any living thing, such as a plant, and using that to create audio. And I put it together for an installation, which I'll play here. And it's not easy listening, but it's an interesting combination of 2 independent sources of audio, one from DNA, one from a living organism, and blending them together as an artistic endeavor. So here it is here. So you can quietly hear that pulsing in the background, which is the DNA audio. And then you've got this droning kind of in the background, which is the audio from the plant. And the combination of the 2, I just chucked them into the mix as a creative endeavor, just to see how it sounds. And that long pitching thing is the plant, and you can see how the plant is here with the electrodes attached to it. It's skip ahead a bit. So here's the electrodes on the plant here, and then that signal feeds into a synthesizer to generate this kind of drone-like musical thing. And I think that's generative music, okay? But I've mixed that with this systematic music from the DNA sequences. And then it just shows the computer here, which is playing the pulsating audio. And just lastly, as a finisher opera, I just kind of, you know, what I've done, for better or worse, is kind of just like pursue the interests I have. Like, I love science and I love doing science stuff, and I love music. And I didn't enjoy doing science during the day and music at nighttime, because I found I didn't have enough time to do both, and each detracted from the other. So bringing the two together, I think, has been kind of a good process for me. And that's taken me to think about music and the science of music, and about how we can apply this to ourselves. And one thing I'd like to do is to start thinking about generating audio from rare diseases. So if you're an individual who has a rare genetic mutation, let's listen to it. And that way you can talk to people about their condition, and use a bit of audio to help understand a bit of the science, just as something that's in the mix there. All right, I think that's it for my slides. So thank you for listening, and hopefully I've been semi-coherent for you. And back to you, Devin.
Thank you, Dr. Temple. That was a really, really engaging and interesting presentation. And I just have to say that they say who hangs around musicians is drummers. I used to be a drummer back in high school, so I believe that we are all musicians. So I thank you for that. We do have one question, and it is coming, forgive my pronunciation, Yadunand, and they're saying, hello, I was wondering how you figured out what note to assign to each DNA base. For example, how did you know to assign C sharp to the first three, etc.? I'm interested in trying to apply sonification to numbers and functions in mathematics, so I was wondering how I would figure out which note to assign to which number.
That's a really good question. And in some ways, it's completely arbitrary. So when you're doing sonification, you're doing this systematic mapping of data to audio. Okay, so it has to be systematic, so that whenever you hear something, it relates to something else. What you choose to use as that note or that instrument is completely up to you. And that's when you can be creative to create something that has science value. So initially, when I was doing this, I just chose a major scale. So a bit of music theory says this group of notes sounds good together. So I started to sonify with the major scale, but then I was scratching my head and I played it to someone, and someone said, hey, Mark, you've sonified the coronavirus genome, but it sounds really happy. And I was like, oh my God, what have I done? And I kind of thought about it. And then I thought, well, let's use minor scales, because minor scales sound a little sadder, a little bit more melancholy. And I thought that was more appropriate for what I was doing. In terms of science audio, it doesn't matter whether it's major, minor, because it's, again, like I said, it's that systematic mapping of data to audio. And I think the other consideration is, well, I chose musical notes. I didn't need to choose musical notes. I could have chosen just noises, just different noises to indicate different attributes in DNA. But personally, I find it difficult to listen to just random, well, not random, but just noises. I need to listen to something that's not going to fatigue my ears. And I thought musical notes was a good thing to do. But when I started sonifying, and if you do some sonification, you might find the same thing, is that people who don't really, the people who just come across your work by chance, they hear it and go, oh, is that music? And then if it is music, then that raises a whole bunch of questions, which is kind of good, actually. But Yeah, so I think the choice is arbitrary and it depends how musical you want to make it. Think about Geiger counters. They're just crackling noises, so they're not musical, but they tell you the more crackle, the more radiation. So there's a clear relationship between one and the other. Each crackle doesn't actually, yeah, anyway, does that answer your question?
I believe it may. They'll have the opportunity to clarify or ask another question in the chat. And this is open for everybody. If you have any questions for Dr. Temple, please drop them now. This is a wealth of knowledge. Oh, and Yadinen, again, forgive me for the pronunciation, said thank you so much. So I believe that answered their question. But if anybody else has any questions, please feel free to drop them in the question and answer chat. Do you have suggestions for how students could experiment with sonification?
Well, I don't know if I've got suggestions of how. I think what I do, like my sonification is heavily dependent on using sort of computer code. And like I've got a little bit of coding ability to sort of say, take this, generate this, take this, generate this, and then to loop it. But I think to begin with, you've got to think about it manually, like, just think about it as a concept rather than think about it as here's the code I'm going to write, because that's quite technical. So I think about the process. So what is the data you're trying to work with? And it can be anything. Like, I think the process of sonification doesn't care what the data was. Okay, you just need to have input data. Okay, so it could be like really trivial or it could be really complex data. It doesn't matter. And then just think about whether the data is whether it has a time component. And if it doesn't, like, for instance, if I took DNA, put it in a spectrophotometer and hit the go button, it would just present me an image, which would be a graph. Okay. Now there's no time on the graph. It's just a graph of, frequent, like it's just intensity versus frequency, it has no time component. So what I would do in that stage, then I'll be thinking, well, here's my data. How do I convert that data into a timeline? And the obvious thing to do would be to start on the left-hand side and look at how the values change as you go to the right-hand side. And then you've got changes in something over time then. So you're reinterpreting the data and then thinking about, well, I've got something that's moving up and down here. How do I map that to whatever it is I want to map? So you could do it manually and you could just get a piano or a guitar or whatever it is and look at the chart and just start thinking about what note do I play as the data changes. And then if you can do coding, then you can sort of take that process and automate it, which is what I've done. But I know people who do sonification and they use it to play to. So they play in real time to the data without creating an audio display of the data. So there's lots of ways you can do it.
I believe, Dr. Temple, correct me if I'm wrong, the website dnasonification.org, does that allow them to sort of test around and create their own strands or enter their own DNA sequence to see how that would sonify as well? So if they want to
Yes, it
toy
does.
around with that. So
Yeah.
I think that is also a way that if you're interested in it, what you can do and how it works. But again, if you want some more clarification, just drop that question in the chat and Dr. Temple will give some more clarification on that.
If you want, I can show you how to do that really
Sure.
quickly.
Yep.
Okay.
And then after that, we'll have one more question and then we'll go ahead and wrap up. So you can go ahead and share your screen. Not yet, Dr. Temple. Not. Oh, did you have a question?
One quick question.
Yeah.
What I'll do, I'll track the YouTube video, and if I see questions in the comment sections, I'll comment in to answer the questions.
That works. Thank you for doing that as well. And I just want to say, everybody who's still in attendance, Dr. Temple is coming all the way live from Australia, so our time difference is really different. So it's pretty early for him, and so I just want to thank you. And we have a comment already saying the same thing. That was a wonderful presentation. Makes one think out of the box. Thank you. So I think we all share that sentiment. And so I just want to give you a great big thank you again, Dr. Temple. And I'm going to share my screen. And then we can, let's see. Thank you so much to Dr. Temple once again, and another thank you to all of our sponsors, including the Claude Moore Charitable Foundation. And lastly, I want to thank you all for joining us today. You will be notified when this recording is posted on our YouTube channel. More information regarding our 2024 webinars will be sent out in the coming weeks. So there will be no webinars in the month of December, as that is a big holiday month. So more information regarding our 2024 webinars when we come back in January will be sent out in the coming weeks. And we hope to see you there as we continue to grow with STEM Lyceums. So again, thank you so much for attending, and thank you, Dr. Temple, for your wonderful information. And like I said, this will be sent on YouTube, and look out in your email for a survey of the webinar for you to fill out. It should take maybe no more than 2 minutes, and that'll be it. So again, thank you, and I hope everybody has a great and wonderful night. Have a good one.